Androgenetic alopecia is characterized by progressive miniaturization. Driven by genetic susceptibility, hair follicles gradually shrink, transforming robust terminal hairs into frail, translucent strands. Clinical data identifies Finasteride and Minoxidil as the primary defenses against this process. These FDA-approved pharmaceutical interventions are proven to slow or reverse the shrinking of the follicle. However, long-term adherence presents a significant clinical hurdle. Patients frequently experience or fear systemic side effects, ranging from scalp irritation to sexual dysfunction, which limits the practical utility of these drugs over a lifetime. This adherence gap has driven widespread clinical interest in dietary supplements. Yet, the evidence supporting these alternatives has historically been fragmented, relying on trials with small sample sizes and a lack of direct comparative data. To resolve this, researchers recently conducted a comprehensive network meta-analysis, aggregating data from 19 separate randomized controlled trials covering 1,658 patients. This network map illustrates the power of the analysis. By charting indirect comparisons through shared control groups, statisticians can probabilistically rank 16 different dietary interventions, even when those supplements have never been tested head-to-head in a single trial. Aggregating this fragmented data establishes an empirical ranking for these pathway-specific therapeutic tools. The relative efficacy of these supplements is dictated by how they interact with the localized microenvironment of the scalp. This flowchart maps the primary androgenic pathway. Testosterone enters the follicle environment and passes through an enzyme called 5-alpha reductase, converting it into dihydrotestosterone, or DHT. When localized DHT binds to androgen receptors in the hair follicle, it initiates a destructive sequence, shortening the active growth phase. That primary pathway is then accelerated by a secondary epigenetic factor, reactive oxygen species, or ROS, triggered by chronic oxidative stress in the scalp. These ROS molecules activate latent TGF-beta proteins. Once active, TGF-beta drives microinflammation directly into the dermal papilla, accelerating the miniaturization sequence. Specific botanical interventions function as targeted inhibitors against these precise pathways. Molecules from saw palmetto extract blockade the 5-alpha reductase enzyme, while tocotrienols neutralize the ROS molecules before they can activate inflammatory proteins. Multi-component formulations are designed to execute these blockades simultaneously across multiple points of failure. These supplements operate as active biochemical inhibitors, disrupting the miniaturization sequence at the cellular level. The network meta-analysis tests these biological mechanisms against the first primary clinical endpoint, overall hair density. To rank the effectiveness of each intervention, the researchers calculated a sucra value, the surface under the cumulative ranking curve. This metric calculates the mathematical probability that a specific treatment is the absolute best option. This chart reveals the results. For increasing overall hair density, the multi-component supplement Nutrafol achieved the highest sucra probability at 80.8%. This formulation produced a standardized mean difference of 0.90 hairs per square centimeter over a standard placebo baseline. Apple extract formulations and pure tocotrienol extracts placed second and third, yielding tightly clustered but slightly lower statistical returns. The statistical performance of the top formulation aligns with its targeted multi-pathway design. The formulation combines saw palmetto for DHT inhibition, curcumin to downregulate androgen receptors, and tocotrienols to suppress oxidative stress. For maximizing total hair count, the statistical data favors these multi-target botanical complexes over single-ingredient extracts. The analysis also applied a second, more rigorous clinical endpoint, terminal hair density. This strictly measures thick, fully pigmented strands, the evidence of a successful reversal of follicular miniaturization, as opposed to overall density, which includes weak vellus hairs. Under this stricter metric, the sucra ranking shift. A botanical complex designated ALRV5XR leads the field, claiming a 99% probability of being the most effective treatment. This intervention produced a standardized mean difference of 1.58 terminal hairs per square centimeter. However, the broader dataset for terminal hair growth revealed a distinct anomaly. The efficacy of these supplements varied significantly from trial to trial. In statistics, this inconsistency is measured as heterogeneity, or I-squared. The dataset showed an I-squared value of over 60%, indicating highly unstable results that required researchers to isolate the hidden variable. This stratification tree reveals the missing variable. In trials where patients combined oral supplements with topical active agents, the heterogeneity plummeted to 25.2%. Oral therapy delivers inhibitors through the bloodstream, while topical serums deliver antioxidants directly through the scalp to optimize the microenvironment. The stabilization of these results suggests that while oral supplements show benefit, the most consistent clinical signals emerge when systemic pathways are paired with localized topical support. We must also consider the demographic characteristics of the patients receiving these interventions. Specifically, the analysis questioned whether a patient's baseline hormonal profile alters the effectiveness of the supplement. To find out, researchers isolated the female patient subgroup and analyzed their specific terminal hair density outcomes. The data reveals a contrast. In the premenopausal cohort, the benefit trended upward without reaching statistical significance. In the postmenopausal cohort, the efficacy spiked sharply, yielding highly significant results. This efficacy spike is tied to the physiological shift of menopause, characterized by a systemic decline in estrogen. Because estrogen naturally opposes androgens, its absence creates a state of relative androgen dominance in the scalp, accompanied by elevated oxidative stress and low-grade inflammation. This creates a much larger reversible space. When baseline oxidative stress is severe, potent antioxidant supplements have a wider margin to generate biological improvements. Precise patient selection is as critical as formulation selection. These therapeutics yield the highest returns in populations suffering from acute hormonal and oxidative dysregulation. To deploy these tools responsibly, we have to evaluate their absolute clinical ceiling. We determine that ceiling by comparing the supplement trial data against both inactive placebos and active first-line pharmaceutical controls. This forest plot demonstrates that against a standard placebo, dietary supplements establish statistically significant superiority for hair growth. However, when measured directly against Finasteride, the aggregate data shifts. The results land squarely on the null line, indicating zero statistical advantage. We must also factor in the limitations of the current data pool. Many of these trials suffer from small sample sizes, publication bias, and a lack of long-term safety tracking beyond 24 weeks. These limitations define the specific clinical utility of dietary therapeutics, providing a statistical middle ground between pharmaceutical dominance and total inactivity. Clinical deployment relies on three data-backed pillars: selecting multi-component formulations based on the specific density endpoint, utilizing topical synergy to stabilize results, and prioritizing demographic subgroups with high baseline oxidative stress. Dietary supplements are not standalone primary therapies, but when mapped accurately to underlying biochemical pathways and precise patient profiles, they function as highly effective evidence-based clinical adjuncts.